I want to introduce Christina Curtis from Stanford, who's going to come up here and talk about the study that we did combining RNA and protein spatial profiling along with bulk expression analysis and think about how to integrate that data set to be able to discover biomarkers. Christina, thank you so much. There you go.
Oops. Excellent. All right, yeah, so it's really a pleasure to share with you some of the very recent work we've been doing in close collaboration with NanoString and the team there to really try and understand how the tumor and immune microenvironment changes through the course of neoadjuvant therapy. And so just to kind of walk us through this, I think it goes without saying that in recent years we've really understood that there is profound heterogeneity both at the genetic, cellular, as well as phenotypic level. And that really this encompasses both tumor cells and the surrounding microenvironment. This, of course, contributes to resistance. And really, as of yet, we haven't fully been able to describe what these interactions look like. And for us, being able to do this in context is really important because, of course, as we want to peer in with greater resolution at the cellular processes that are operative, we also want to do so with an appreciation for where these cells are, both with respect to tissue architecture, but the rest of the microenvironment. And so our work in breast cancer dates back some years now, and I just point out this slide to say that much of what I'm going to talk about today is focused on HER2-positive disease. This is one of the prior to the dawn of trastuzumab, the worst risk of relapse subgroups. We can see that the risk is quite high, and this is in a cohort of nearly 2,000 women where we have very rich clinical follow-up data. This is, of course, now outdated, and we wouldn't see the same pattern today. But we still don't fully understand the drivers of resistance to some of the archetypical targeted therapies that have been developed to treat this disease. And we know that trastuzumab has significantly improved outcomes. This is both true of the early stage setting as well as advanced HER2-positive disease, and really has become the mainstay of clinical care for this patient subgroup. There's a number of HER2-targeted agents, and this is in fact why we set out to start to address these questions in this system, because we knew that we could evaluate not only one agent, but maybe multiple agents and the mechanisms that are potentially associated with resistance. Of course, trastuzumab being one of them, but other targeted therapies as well, such as lapatinib. And this is really the focus of the trial that I'm going to describe to you today. There are many known components of the signaling pathways that are operative downstream of HER2. Again, as Nero pointed out, this gives us actually quite a bit of information on what ground truth we might be looking for in terms of pathways that are altered as well as compensatory signaling alterations. And so it's also really well appreciated that there are manifold mechanisms of resistance. And by this, I've just highlighted a few that we do see here. There are many others, but I think a big challenge, and certainly what we've observed through largely genomic studies of this process, is that there's heterogeneity amongst patients in terms of what those mechanisms are. And in fact, while we have a number of candidate biomarkers, this is not universal across patients and really motivates a deeper dive into not only the genetic factors, but the signaling and potentially immune factors that contribute to this. So we've been interested in studying how HER2-positive breast tumors evolve through the course of neoadjuvant therapy. Much of our work to date has been based on looking at this from a genomic perspective, but I want to point out that the neoadjuvant platform is really powerful for these kinds of studies because essentially we're going in and obtaining a diagnostic or pre-treatment sample. The patient then undergoes a defined period of treatment, typically speaking in combination. So we have trastuzumab in combination with chemotherapy. And then at the time of definitive surgery, we know whether or not this patient has achieved a pathologic complete response. This path-CR is also associated with outcomes. And so this gives us a really short-term readout of the mechanisms that might be associated with response. So in this study, we were interested in really taking this to the next level and not only looking at what happens from a genetic perspective, which we already have some data on, but really to be able, for the very first time, to go in and ask what is happening within a spatially resolved context to the tumor and immune microenvironment. And of course, these are FFPE samples. Many of them involve core biopsies. So we're, in this case, obtaining a core biopsy not only from the pre-treatment sample, so we refer to this as baseline, but also on treatment. So this is a mere 2 to 3 weeks where the patient has received targeted therapy alone. And that's really important because it means that with this kind of approach, we're able to disentangle the contributions of the targeted therapy from the chemotherapy. Afterwards, the patient goes on to receive chemo as well, and then we get a third sample if the patient did not respond at the time of definitive surgery. So we're working with really precious material, and in fact, the study that I'll describe today, led by Catherine McNamara, who's an exceptionally talented MD PhD student in the lab, focuses in on a trial. And so this is through a collaboration with Sarah Hurwitz and Dennis Slayman at UCLA. We focused in on the TRIO-B07 study, and as I said before, we obtain a pre-treatment biopsy at time 1, an on-treatment or run-in biopsy at T2, and then at the time of definitive surgery, we obtain a surgical sample. Of course, if the patient has responded well, we anticipate that largely what is left is going to be infiltrating immune cells and very limited tumor cells. And that's, again, an important reason why being able to profile this in context really lets us make the most of these very precious samples. So we profiled a total of 28 patients from this cohort. They're roughly equally distributed amongst responders, those that achieved a path-CR, and non-responders, those that did not. So we took the GeoMx largely IO panel, but we did customize this really because we had in mind a number of questions that revolve around the signaling pathways that we know are operative here. And so we have, you'll see on this, both markers of sort of tumor cell existence and proliferation. We added in and enhanced the sort of HER2 signaling pathway members because we felt that this would give us a handle to some extent on ground truth and what's really going on here. And then there's a large number of markers of immune response and negative immune regulators. In addition, we went in and used one of the early RNA probe sets. So this is essentially a 96-plex IO panel, and the probes that are shown here in orange are those that have corresponding protein probes. So we can see there's a lot more on the RNA panel, but we believe that this gives us an opportunity to really ask the question, what is the degree of correlation between RNA and protein? And moreover, it expands out into some additional markers that are of interest to us from both a signaling and immune perspective. So our approach to selecting regions of interest was to go in and use a pan-cytokeratin-based marker. You can see these are actual samples from our cohort where we're staining for CD45 as well as dsDNA. And we refer to these as tumor-enriched regions, the pan-cytokeratin-positive regions, or pan-cytokeratin-negative tumor microenvironment enriched regions. And what we can see just from a quick glance at the RNA level is that we're actually finding what I think is really quite encouraging. So what I'm showing you here is simply the counts. These are housekeeping normalized for individual ROIs. And for each ROI shown here, you have the pan-cytokeratin positive next to the corresponding pan-cytokeratin negative region. You can see that by and large, we see a pretty striking enrichment for known immune markers that are in the pan-cytokeratin negative region. And you can see the actual staining from these tissue samples over here. We're looking at a series of baseline samples, run-in, and so forth. So we found that, overall, this was really quite encouraging, what we anticipated to see. And, moving on from this, really, we had a number of questions. First of all, at a very global level, we kind of wanted to understand how far we could push the limits of this technology to understand what spatial heterogeneity looks like in context, and really during the course of neoadjuvant therapy. We were interested in how the tumor immune microenvironment changes with treatment, and in particular, what are the changes that occur in the HER2 signaling pathway? And can we really parse out what's happening in the responders versus non-responders? Might we be able to pick up additional compensatory signaling pathways that could contribute to resistance? So we had a number of sort of sanity checks when we first got a hold of these data. Some very fundamental questions that, believe it or not, actually haven't really been addressed. One of these is simply how consistent are HER2 protein levels when sampled from different regions of the same patient's tumor. So you're seeing a representative case here, patient 39. These are baseline samples. So this is a baseline taken, prior to treatment, and we're looking at different regions of interest. We actually see that the levels of HER2 are fairly consistent. These are log2 normalized values. We can do the same thing, in fact, and now ask the question, how variable is this not only within patients, but across patients? Showing another representative case below this, patient 69 again at baseline. And encouragingly, we see that really the HER2 protein levels are far more consistent within ROIs from an individual patient. We're comparing across four different ROIs, and they are elevated, but really not so very dramatic between different patients. And I think that, when we actually think of the wealth of IHC data, there is considerable evidence to suggest that HER2 is actually amongst the most stably expressed in these HER2-positive patients relative to other protein markers. And we can quantify this really, and we see that, again, the mean square error just capturing this variability is higher across patients than within patients. But in fact, this is true that really when we look at run-in, we see a pretty dramatic increase. So once the patients have actually seen therapy, we see a striking degree of variability in their HER2 protein expression levels. We can quantify this not only looking at HER2, but really looking at what is the variability across different tumor regions, shown here for the baseline at the top, the run-in tumor region, and then followed by the microenvironment regions at baseline and run-in. And what we see when we look at this at a protein level, just following on this histogram here, is that there's actually substantially increased heterogeneity across all probes at run-in. And this is pretty dramatic here in the tumor, perhaps more so than in the microenvironmental regions. We see that this is also true when we look at an RNA level, and really that we just see this dramatic increase in heterogeneity. And I think it's really nice that we're able to, with these very precious samples, go in and ask this question across not just a few markers of interest, but many more, and really to have a more global perspective of what these changes look like. So one of the key questions that we wanted to ask is really, what are the major protein changes that occur during targeted therapy? And so just to orient you, these are volcano plots. What we're looking at here along the bottom is the negative log fold change relative to the sort of strength of significance of these alterations. And what we see is that in patients that do achieve a pathologic complete response, after just one cycle of targeted therapy alone, so this is 2 to 3 weeks on targeted therapy without chemo, we see a really dramatic decrease in known HER2 signaling pathway components, as you can see from over here. Now, we can look at this from another lens, and in fact, if we look at the alterations that are largely downregulated, what we see is that there's a really striking enrichment for immune signaling at the time of run-in in these patients. So we're seeing a decrease in HER2 signaling, an increase in immune signaling, and we're able to do this within context across individual patients, and these trends are, in fact, quite statistically significant. We can also really now ask the question, what's different between responders and non-responders? And so shown on the left, again, these are the responders. On the right, you'll notice that, first of all, the thresholds have changed. So really, the axes labels are significantly different here and much lower, and there's a far less noticeable trend with respect to the change in protein signaling as well as immune response in the non-responders. However, that being said, so here we're really looking at this from a temporal perspective, and what we can see is that the patients that don't achieve a pCR at the time of run-in, while there are very minimal changes, by the time these patients do go on to surgery, we actually see that there is a reduction. It doesn't reach the same levels that we see in the responders, but we do begin to see this certainly reduction in HER2 signaling, Ki-67, and a more modest increase in immune markers. So really, overall, what we're finding is that there's a far more dramatic increase in immune response and decrease in cancer signaling and this is true not only at the protein level, as I've shown you here for the past CR cases previously, but this is also recapitulated at the RNA level. And so we, in fact, can see overlap in terms of some of these markers. And as I said before, the RNA panels quite a bit more expansive than our protein panels, so we're picking up additional markers that we didn't see at the protein level. And of course, really, I think this demonstrates, to some extent, a tight degree of correlation, but this is not universally true. And of course, this could be due to both biological reasons as well, and that's something that we're interested in exploring. So really, what I've shown you is that the GeoMx platform allows us to make multiplexed and spatially resolved measurements in very precious archival tissue. We're able to delineate contributions from the tumor cells as well as from the immune microenvironment. And here we've really used this technology to look both at protein but at RNA levels in HER2-positive patients that have been treated neoadjuvantly, and we're able to monitor these changes during the course of therapy. We see that HER2 levels are more consistent across regions from an individual patient than they are between patients. We find that tumor heterogeneity increases quite profoundly during the course of neoadjuvant therapy. And because we seldom obtain these run-in biopsies, we're really looking into new biology that's not been previously understood. And of course, there's a lot of hope that if we can start to sample these patients earlier, before they've gone through a whole course of therapy, perhaps we can intervene and identify those who will respond, or alter the course of treatment for those who won't. We see this massive reduction in HER2 signaling that occurs with targeted therapy, really dramatically. This is taking place within just 2 to 3 weeks on targeted therapy. And these changes are far more dramatic in patients that do go on to achieve a past CR. And I think we're really interested in ongoing work are trying to disentangle the various contributions and the temporal aspects of these changes in responders versus non-responders, and to link the RNA and protein data. And with that, I'll thank the people in the lab who have done this work. As I mentioned, Catherine McNamara has really led this. We have fantastic collaborators on the clinical side, and really the team at NanoString has been fabulous in really generating this very comprehensive, large data set that's allowing us to peer into these tumors with unprecedented resolution. So thank you.
Thank you, Christina. I'll take that. Any questions for Christina? Wait till the mic comes in, and then we'll be good.
Thank you. I was just wondering, the differences that you found based on the spatial context, do you think it's plausible to identify some of those biomarkers and then take it back out to a bulk, or do you think that the spatial context is really key in identifying this after the discovery phase of the study?
Yeah, it's a great question. And I didn't show data. We've done bulk profiling of all of these tumors at an RNA level. Certainly, there are signaling pathway components that correlate very tightly with the bulk data. There's also changes that we don't see correlating with bulk that we find only when we do this from a spatial perspective, and I think that's going to be important to really drill into. Some of these look very, they're very much implicated in apparent responses. So I think that it's the usual challenge, and that we have these admixed populations, and we don't really know what we're looking at. So I think some of them, if they're dominant, we certainly should be able to recover them in the bulk, but that's not always going to be the case. And I think for deconvolving the contributions of the tumor and immune, this is particularly important because it's very difficult to do that sort of algorithmically from the bulk expression data.
Thank you.
Great. Any other questions for Christina? No.
Hi. Could you comment on whether you have used other types of high-dimensional profiling techniques on these samples, such as single-cell RNA sequencing or CyTOF? And then, if so, how does your result from this particular platform correlate with those other technologies?
Yeah, unfortunately, these are really precious samples, so we don't have the luxury of running them through kind of every assay that we might like. And I think that for us, really, it was a priority to be able to not dissociate the tissue for these kinds of analyses, in part because we did really want to be able to look, and I think in this sort of short presentation don't have time to get into the details of what those topographies look like, the interrelationships between different cell types. So we don't have those data, we really only have the bulk other types of genomic profiling.
Any other questions? I had a couple for you, Christina. One was, you were probably one of our early collaborators on the breast cancer side, because when we looked at sort of the break in immunotherapy, most of the work was in melanoma or lung, some of these areas that have huge immune components, but more and more now it seems like there's a lot more interest in looking at immune responses in tumor, in breast cancer samples. And what's the change in the trend that there's more importance or looking at spatial information and immune response in breast cancer, you think, now more than before?
Yeah, I mean, currently that's certainly being driven, I think, on the triple-negative breast cancer side. And obviously there's a lot of questions surrounding whether we can make, some of these tumors are already perhaps immunologically hot, some are cold. What is it that defines those that are cold? Are there ways that we can, from a clinical perspective, prime them so that perhaps they are hotter? And I think, there's just a lot to be learned about what those architectures look like, because we've not been able to peer into the tissue in that fashion. So I think there will continue to be interest in resolving those questions.
The other one I had was, I think we pointed out that yours was one of the largest studies we did, and we learned a lot in terms of how to look at large sets of spatial data sets. I'm curious about what are the things that you took away as learnings in terms of the data that you saw, how you analyze it, when you start to look at how the spatial information is organized. What are some of the learnings you took away from that?
Yeah, so I think, there's study-specific considerations for how you go about masking and what those, priorities are. Obviously, there's sort of global features that can be used to try and identify tumor versus immune cells. I think some of this may also come from, the specific questions that are at hand, whether you want to define a particular macrophage population or not. And so I think, the user has a lot of freedom in how you could customize those questions. And for us, this was really, I think, a first glance, a first learning of how we could go about doing that. And also really want to try and avoid biasing the analyses too much, given how very little we actually understand about tumor immune architecture. So, yeah.
Great. Well, thank you so much.
Sure.
Thanks so much, Christina.